All right, the last system we are going to discuss is the nervous system. So the basic functions of the insect nervous system is to receive stimuli from the environment, integrate sensory data, and coordinate information to create behavioral changes. Now note that in insects, this coordination is limited compared to humans. So in humans, we have a centralized nervous system, which means that all of the control for our nervous system comes from our brain inside our head. But with insects, it's decentralized, which means that the main coordination systems are not going to just be found in the head, but actually throughout the insect's body. So there's two major components to the insect's nervous system, the brain and then the ventral nerve cord. The ventral nerve cord is composed of a series of ganglia, or small nerve masses, or mini-brains. So as I mentioned, the ganglia are a bundle of nerve masses. So this picture shows an example of what the insect nervous system would look like. You've got the brain in the head, and then the ventral nerve cord with the ganglia going throughout the rest of the insect's body. So the insect's brain, which is located in their head, is trilobed. So it contains three different sections: the protocerebrum, which controls their vision, the deuterocerebrum, which controls their antennae, and the tritocerebrum, which controls their labrum, or that upper lip section. Then after we leave the brain, we enter the ventral nerve cord region. The first ganglion that is found in the ventral nerve cord controls the feeding center, or the rest of the insect's mouth parts. So the next set of ganglia are the thoracic ganglia, which control movement. So each set of the three thoracic regions will have some of these nerve masses, and each of these masses will control the movement of those individual segments. Then we have the abdominal ganglia, which control digestion, excretion, and reproduction. Because of this unique arrangement of these nerve masses, insects can do some really remarkable things. One example is with the praying mantis. Now, the praying mantis, of course, is predatory, but it couldn't care less if it's eating a beetle or another mantis. This, of course, makes it tricky for the male mantis to mate with the female and not be eaten. So this tends to be how things end a lot of the time. So what you'll see is the male will jump on the female's dorsal surface and begin mating with her. He is readily pumping sperm and trying to do it before she has a chance to notice he's there. Sometimes he'll also present her with a prey item to preoccupy her, but if she does detect that he's there, then she may bite off his head. And you might think that this would stop the mating process, but in insects it does not. Even though the male's head has been removed, he will still continue to inject sperm. She can then eat the thorax, and he will continue to mate. Again, this is because of the unique characteristics of the nervous system. It has nerve masses controlling the segment that is used for mating. So until the nerve masses for that segment are impacted, that male mantis will continue mating. The final thing we need to talk about now is nerve impulse transmission. When we talk about this, basically the insect's nervous system, like humans, is composed of neurons, and you have these series of neurons. And in between these neurons are gaps called synapses. Basically, you have neuron 1 and neuron 2 with a synaptic region in between. Well, there is a problem with these gaps. Because if there is a neuron pulse transmission that travels along neuron number 1, it needs to get to neuron number 2 in order to make some type of response. How does it get across it if it has this gap? Basically, it needs a type of transmitter substance, and in the case of insects and even humans, this is called acetylcholine. You have the nerve transmission traveling down neuron 1. It reaches this gap, so produces acetylcholine. This chemical transmitter then carries that impulse from neuron 1 to neuron 2. Once acetylcholine carries that nerve impulse from neuron 1 to neuron 2, it's really important that that chemical transmitter is destroyed. If it is not destroyed, you can have continuous firing of that nerve impulse, which can be very dangerous to the insect. So an enzyme is produced called acetylcholine esterase, and it destroys the acetylcholine. So what happens if this enzyme is not produced? Well, it can cause a continuous nerve transmission, as I mentioned, or that continuous firing. And so Knowing this, it's been used as a mechanism for some insecticides because if this nerve transmission is not stopped, it can turn out to be fatal for the insect. So what are the implications for humans as far as using these insecticides? Again, humans have a similar nerve impulse transmission process as insects, and these insecticides are meant to prevent the acetylcholine esterase from being produced. Therefore, the insecticides can affect our nervous system in the same way. That's why it's very important to read the label and use these insecticides responsibly. Now, of course, it's going to harm us at higher levels, but it's just very important that we follow those directions carefully. Now again, because our nervous systems act in similar ways, we can use insects in a variety of research studies to understand more about the nerve impulse or nerve impulse transmission in humans. For example, here is a study where they used spiders and wanted to test the impact that different drugs had on the nervous system of these spiders. So on the left-hand side, there is just the normal spider web. In the middle, it was a spider that was exposed to a high amount of caffeine. And then on the right-hand side, it was a spider that was exposed to LSD. So you can see how the different drugs have had an impact on the construction of these webs. So they've done these studies in order to understand how the drugs affect the nerve impulse transmission process and then try to relate these back to humans. There have also been studies looking at the impact of alcohol on honeybees because on a molecular level, the brains of honeybees and humans work the same. So knowing how chronic alcohol affects genes and proteins in the honeybee brain may help us eventually understand how alcoholism affects the memory and behavior in humans. So with that, we've concluded our discussion on the first 2 points listed here, and the final thing we need to focus on is comparing the similarities and differences between the internal anatomy of humans and insects. We've focused on 5 basic internal systems: the digestive system, the excretory system, the circulatory system, the respiratory system, and finally the nervous system. So after listening to these lectures, you should be prepared to discuss the similarities and differences between the physiological systems of insects and humans. What I would like you to do for this assignment is identify at least 2 similarities and 2 differences between insects and humans for each of these systems. You can find more information on Canvas, and as always, you can email me with any questions.